I'm going to introduce our speaker today. Olivier Saux is a researcher that has worked with PXE International for quite some time, since the beginning, really, almost 25 years. He's responsible for finding the gene with us. When he was working in his lab in Hawaii, he was able to isolate the ABCC6 gene for PXE. We have worked with him ever since, supporting him in his work and supporting his students, and we are very happy to have him with us today.
All right, thank you for the introduction. So I'll be sharing with you today my latest results that have been now published in that regards the ABCC6 pathway and dietary phosphate in calcification. So the picture that you have here is the medical school where I'm working at the University of Hawaii, surrounded by beautiful palm trees. But I thought I would begin by giving you an introduction on biological calcification, which is actually a rather complex process. So there are two types of calcification, obviously. The physiological side here, I can move this pointer, the physiological calcification, which is essentially what you would normally find in your bones or your teeth. And there's also the ectopic calcification, which is an abnormal calcification. Here are some definitions, some of the words I'm going to be using. Ectopic calcification, which I just mentioned, is essentially a mineralization of soft tissue that occurs in the absence of any systemic mineral imbalance. When everything looks normal, there is this abnormal calcification that occurs in places where it's not supposed to happen. The dystrophic calcification is a form of ectopic calcification, and that is essentially generally in response to a specific injury. There could be a lesion, and I will be mentioning a dystrophic cardiac calcification phenotype a little later. Metastatic mineralization is a different type of calcification that occurs, but this time when there is an elevated presence of calcium or phosphate, either because there is a problem with absorptions or there's a problem with secretion. And that doesn't really concern us as far as PXE is concerned. So ectopic calcification can affect multiple types of tissue, soft tissue, such as those affected in PXE, but also cartilage tissue. And the primary concern in the general field of medicine and biomedical research is cardiovascular calcification. The heart itself, the myocardium, can calcify after injury, such as a heart attack. That has been documented. It's not very common, but it happens. Otherwise, the main type of cardiovascular calcification that exists and is concerning from a medical point of view is the valve in the heart or the blood vessels, the vascular type of calcification. And the vascular type of calcification, we distinguish two types. The media, which is essentially the thickness, the wall of the arteries, or the intimal calcification, which is the inner portions of the arteries. That inner portions of the artery generally is the calcification is associated with atherosclerosis, the deposits of cholesterol and the buildup of cholesterol in the artery. That generally leads to a certain amount of calcification, which is associated with risk factors. When it comes to calcification in the thickness of the arteries in the wall, then there are genetic risk factors such as PXE, for example, or generalized arterial calcification of infancy, which I'll talk about later, beta thalassemia, calgia, and there are many other diseases associated with this. And they also acquired risk factors that are here are such as chronic renal insufficiency, which is a metastatic type of calcification, diabetes, aging, and other conditions that are not related to any particular gene. So studying PXE or GSCI is an interesting for itself to solve and understand this disease, but I forgot to mention here, the type of calcification we're seeing, whether in the bones or the ectopic calcification, the abnormal calcification, is essentially hydroxyapatite. The majority of the calcification is hydroxyapatite, but that's not the only one. And you can see from the formulation here, it is mostly calcium and phosphorus. And it's the same calcification, same hydroxyapatite you find in your bones or the teeth, or in the ectopic calcification in the skin, the eyes, or the vasculature in the case of PXE. So going back to the genetic risk factor that I was mentioning, studying PXE and these diseases, It's important for understanding those diseases and finding solutions for them. But what we can learn there, we can also reapply to the other conditions, such as the acquired risk factors or atherosclerosis. So studying PXE has benefits well beyond PXE. So again, some of the basic information regarding calcification and regulation of calcification. Calcium and phosphorus, PI here, have really many essential biological functions, such as in the cells, such as cellular signaling and how the cells communicate and regulates its functions. It has function in energy expenditure in the cells, etc., etc. And at normal physiological concentrations, both calcium and phosphorus are readily, they're near saturations and they're ready to crystallize. It doesn't take a whole lot of effort essentially to precipitate and form mineralization. So this is good because it makes it easier to make bones, but it's actually pretty bad for soft tissue. So the body has to come up with something to balance this. Right? We want to have calcification in the bones, but we don't want to have calcification in soft tissue. And there are many things that regulate this balance. The kidneys functions, the endocrine system, your diet, and of course, as the diet is absorbed through the gastrointestinal tract, the GI here. And all of this influences the amount of calcium and phosphorus that is present in the body. Currently, in our body, most of the calcium is locked up in your bones, 95%. The majority also of phosphorus is locked up in the bones in the form of hydroxyapatite. And phosphate, which has this formulation here, you can see it has 2 phosphorus and a bunch of oxygens around it. This phosphate is the main inhibitor of soft tissue calcification, whether the valve, the muscles, the artery, the skins, all the tissue that I mentioned earlier. So that phosphate, again, is the main inhibitor to prevent calcification from occurring in soft tissues. However, body is also well equipped, specifically when it comes to bones, to prevent phosphate from stopping the mineralization in the bones. So therefore, the bones is equipped with a series of enzymes called alkaline phosphatases, which actually breaks down phosphate and use the phosphate broken down in order to actually build calcification in the bones. So the body is really well equipped to construct and maintain its bone and prevent calcification in soft tissue, primarily using phosphate as its main inhibitor. So diseases, aging, tissue injury could have an impact on all this function and therefore calcification and phosphorus concentration levels and mineralization. Of course, this is just not this. There are many genes that are implicated in the inhibition of calcification and its regulations. ABCC6 is on top of the list because of our interest, but there are many other genes involved in the inhibition of calcification. And there are, of course, a number of genes that promote calcifications in bones in particular, and these are listed here. So it's a really complex system. So ABCC6, which is the proteins that is mutated in PXE and the gene has mutations, and it's a transmembrane protein. So here I'm going to be talking about what this protein is doing and how it leads to PXE ultimately. So this is a schematic of a cell here. This is a typical image you find in the textbook of a cell. You have a nucleus, various organelles in orange and blue and green here in the cell, but more importantly, the cell is surrounded by a plasma membrane, which is here. And that's this gray envelope surrounding the cell. So if we zoom in a little bit here and look at one slice of this, you get something like this. Right? So this is a bilayer of lipids, and you can see in this lipid there's all these blue structures here, which are actually membrane proteins, and they have multiple types of functions. Some of them are channels proteins to let things in and out, and you have also the function associated with this thing. And so one of those blue things is actually ABCC6, and it looks like this. So you have the plasma membrane here and this yellow color, and ABCC6 is stuck in it. So you can see this is the transmembrane area. That's why protein is called a transmembrane protein. And most of the protein, in fact, is inside the cell. There's just a little bit of it that sits outside the cell, but most of it is within the inner portion of the cell. And what is it doing? Well, it's actually transporting something outside of the cells. And For the time being, we're thinking that it's actually pumping or effluxing adenosine triphosphate, or ATP. How it does so, it's not quite clear, but we know the effect of ABCC6 is essentially to release ATP from the cell. So for the time being, we're thinking that ABCC6 is essentially pumping ATP outside the cell. So this is the structure of ATP. It's a little complicated here, but the important part of ATP is the P here, with the cursor here. Okay, the T represents the triphosphates. There's 1 phosphate, 2 phosphates, 3 phosphates. So it's a triphosphate molecule. And if you cleave off 2 of the 3 phosphates, that generates pi phosphate, which is a diphosphate. So here's the connection now. Well, ABCC6, as you see the title here, is linked to pi phosphate production. And this is what we think happens. We have good reason to think that this is what's happening, but there's always some obscure aspect of it that we still have to study. So the first thing, you can see a question mark next to this ABCC6 proteins in blue here. So this is the protein ABCC6. And as I mentioned, it seems to be effluxing or pumping out of the cell ATP. The question mark here represents the unknown aspect on how it actually does this. We don't fully understand this. But as I mentioned earlier, this ATP is degraded once it's outside the cell very quickly by an enzyme called ENPP1. It's an ectonucleotidase, and it will actually break down phosphate, as I mentioned earlier, into this inorganic phosphate, PPI. And the rest, what is left over from the breakdown of ATP, is adenosine monophosphate. This phosphate, once it's broken down and separated from ATP, then it's essentially taken up by this blood circulation into the bloodstream. And this is what prevents soft tissue calcification throughout the body. So this primarily happens in the liver. There's also some expression of ABCC6 and ATP generation in the kidney, and perhaps other tissue as well. But the majority of this happens in the liver. The story doesn't stop here. This adenosine monophosphate does something. It's actually further degraded by another enzyme. It actually generates adenosine and inorganic phosphates. And this adenosine is actually, it's a little complicated here, but this adenosine, through a receptor at the surface of the cell, will prevent the synthesis of this enzyme called the tissue non-specific alkaline phosphatase. So this is an enzyme that otherwise, when it's synthesized, it would actually degrade a pi phosphate into inorganic phosphate, which activates calcification. This is the sort of thing you would see in bones, for example. So degradation of pi phosphate leads to inactivation of calcification. So this molecular pathway that involves ABCC6 at the beginning here is essentially geared and set up to generate pi phosphate, which ends up in the bloodstream. But importantly also, the second part of the pathway is making sure that TNAP, this tissue non-specific alkaline phosphatase, is not synthesized and therefore will not break down pi phosphates. On the one hand, we're making sure that we're synthesizing pi phosphate. And on the other hand, at the end of this pathway, we're making sure that it's not being degraded. So this whole pathway makes sense as we're seeing right now. So I kind of represent this pathway at the surface of one cell, but the reality is a little more complex, of course. And this left portion here outlined in this pinkish color here mostly happen on the liver, on the hepatocytes, so the cells of the liver. And the rest of it, the right-hand side here outlined in blue, happens in other types of tissue. So this representation of the pathway here is really useful to understand how things are working. But as I said, it's a little more complicated because it's not happening on the surface of one cell. It's kind of broken down between various cells and tissues. So in this pathway here, we have ABCC6 initiating this pathway, and that's the enzymes and the proteins that leads to the development of PXE when it's mutated and no longer functioning. The other enzyme in this pathway, ENPP1 here, is also leading to causing another disease with calcification called generalized arterial calcification of infancy, and I'll mention some of the specifics of that disease in a moment. And the third enzyme CD73 here is also causing another disease with calcification that is similar to PXE. So again, if this whole pathway makes sense for the generation of high phosphate and its inhibition of its degradation, it also makes sense when we're looking at the pathology associated with these various proteins, ABCC6, NPP1, and CD73. So you're all, of course, very familiar with PXE, so I'm not going to linger too much on this slide. Here, it's primarily caused by a mutation in ABCC6, but there are also PXE patients that only have ENPP1 mutation. The generalized arterial calcification of infancy is primarily caused by mutation in ENPP1, although there are some patients that carry only ABCC6 mutations. That speaks to the fact that these two enzymes are really having similar, these two proteins, so we have very similar function. GSCR is essentially a PXE on steroids. It's very severe calcification that affects cardiovascular tissue, and it's early onset, means it happens very early, generally during pregnancy, and it has a very high mortality, unlike PXE, and the mortality occurs within the first year of life generally. If PXE is rare, this disease is even more rare. There's only a few hundred cases that have been reported so far throughout the entire world. I'm not going to linger too much on this, but this is actually this black thing here represent the calcifications in an arteries in a patient. These white lines indicated by those arrows here are calcifications in the aorta, and there's also periarticular calcification here in those images. The other disease, which used to be called ACDC, but there's now another name for it, it's called calcification of joints and artery. It's pretty descriptive, and it's associated with the absence of adenosine-mediated TNAP repression. If you remember, TNAP for the pathway. So TNAP, instead of being repressed normally here, is actually allowed to be synthesized. And when it's synthesized, it will actually degrade pyrophosphate in particular. And the patients having mutation in these genes develop periarticular calcification, as you can see in these images in C and D. But here on the far right, you can see this massive vascular calcifications in the leg of those patients. So the symptoms of these three diseases, CALDIA on one hand, GSCR on the other hand, and PXE are really similar. And they are all part of the same pathway, the pathway that is here. So what we've learned in the last few years is that ABCC6 functions regulates plasma pyrophosphate. And so what we see in particular in PXE is a pretty large deficit in the amount of pyrophosphate that circulates in the blood. We've seen this in patients, so this is the first report showing that Dutch PXE patient in particular had a severe deficit in pyrophosphate in the blood. There is essentially about 40% of what is present in healthy individuals. And these investigators in 2014 also show that in the mice that have no ABCC6, and knockout mice, there's also a significant depletion in the amount of pyrophosphate that the liver puts out into the bloodstream. So this was done through a liver perfusate experiment, which is here. And this is a small description of what this experiment looks like. It is actually meant to look at specifically the liver, where ABCC6 is mostly expressed. And so you isolate the liver in a mouse model by tying off the upper inferior vena cava and the portal vein at the bottom. And then you inject a buffer to clear off the blood from the liver, and then collect that buffer into a test tube here. And then you can measure what's coming off the liver by measuring what's in the test tube here. So this is what this experiment has done here by measuring what's coming off the liver specifically without the rest of the body. And you can see in the mice that don't have ABCC6, there is a lot less pyrophosphate being produced by the liver. This has been confirmed by other studies. This is a French PXE cohort showing the same thing. There's a lot less pyrophosphate in the plasma and the blood of the patients, or also in the blood of a mouse model. Same thing, it's an ABCC6 model. So this mouse model is here. I'm just going to introduce that mouse model to you. The one I'm using was created in Amsterdam about after Bergen in 2005. This paper was published, and I've been using the mice ever since. you can see this is a picture of the mouse. They are gray or white most of the time in our lab here. And these mice will develop a phenotype that is fairly similar to the PXE phenotype. Of course, a mouse doesn't have the same skin as humans, so they don't have the same type of calcification here, but they do have calcifications in the vibrissae, in the whiskers, right here. And this is what you see in this image here on the bottom left here. This is the vibrissae, and the capsule of the vibrissae calcifies. And this staining here, this red, brown red color, shows the calcification, the abnormal calcification in the capsules of the vibrissae. The mice will develop also arterial calcification. This is the intimal calcification. This is typical of elastic fiber calcification, and it's also fairly similar to what we see in human PXE patients. Mice have a calcification in their eyes also. This is this big asterisk represents massive calcification in the retina, where we expect it to be, and it's very similar to what we see in PXE patients as well. The mice develop an additional phenotype, which has not been described in humans, fortunately, but it's very useful to study calcification in connection with ABCC. And it's called a cardiac calcification phenotype. In fact, it's called a dystrophic cardiac calcification phenotype. And this happens in response to injury. Here, this is the bottom portion of the heart that has been injured, and you can see this white deposit here, which is essentially calcification that occurs where the injury was induced to the heart. And I will be actually using this phenotype, this calcification, to study ABCC6, and I'll mention some of the data here. So the etiology of PXE, which is the origin of PXE, relates to a deficit, again, in pi phosphate. So it's really obvious now that that's really the case, and that's the reason for the calcification in PXE. So an obvious treatment, once this paper was published 5 years ago, was immediately, we thought that maybe supplementation would be really a good idea. How about we add pi phosphate to mice or give pi phosphate to patients, and we see whether we can actually alleviate and suppress calcification. This has actually been shown with, to be effective with other disease models as well, which is listed by those publications here. So we tested whether the administration of high phosphate to our mouse model, ABCC6 knockout mouse, could rescue the chronic calcification phenotype, which is the human analog, what we see in the skin. So this is the calcification in the vibrissae in this particular case. We can easily quantify and measure it. But we also looked at whether we can reduce the dystrophic cardiac calcification that I just explained a few minutes ago. So this calcification phenotype in the heart. We did both because this is the human analog for PXE, but this takes months to study in mice. The dystrophic cardiac calcification phenotype here is useful in the sense that we can actually study this within a week. So in that sense, we generally approach both calcification when we study PXE mice. The way we decided to do this was the simplest way for the time being, which is, or at least when we decided to do this, we injected pi phosphate to the mouse rather than giving it to any other means. So, and then we use an intraperitoneal injection, which is represented by this cartoon here. We injected it essentially in the belly of the mice. And this is how we've done it. So we decided on a protocol before we embarked on the study. The first protocol was associated with the heart calcification. And we did this over a period of 8 days. So the day before we induce a cardiac injury, we start giving the mice a single injection of high phosphate. We induce the injury, which is marked here by DCC, which is a dystrophic cardiac calcification phenotype. So we induce this cardiac injury. And then we give daily injection of high phosphate to the mice all the way to the 7th day here when we harvest the tissue. We injected the mice daily, and we use various concentrations, which are here. And of course, when you do an experiment like this, we need to have a control. This is the baseline, and we injected saline instead of high phosphate. And saline is essentially a salty, a salt solution in water. High phosphate has sodium, so it was a good control because we injected saline, which contains sodium, but no high phosphate. So in these control experiments, we did exactly the same thing, but instead of injecting high phosphate, we injected this salty water solution. And again, this is the cardiac calcification. The other things we wanted to do is the chronic calcification, so the human analog to the calcification, so the PXE manifestations in the mice. Because these phenotypes occur over a much longer period of time, we decided to do it over a 5 months period. So the mice are born, we let them weaned. Once they are weaned from their mother, we start injecting at about a month of age a phosphate. And we did an injection daily for 5 months. This is to see whether we could stop calcification from actually developing in those mice. So we let them go for 5 months of age with daily injection. Why we use 5 months is because before we studied the calcification, the development of the calcification in the whiskers, in the vibrissae here. And this is, you can see the growth of this calcification. And you can see that at 5 months of age, about here, we're in the middle of this curve where calcification seems to be the most active. So it looks like to be a good endpoint to define the effect of high phosphate on calcification. Now, the other things that we need to do is, if we are going to treat patients later on, there are going to be patients that will be treated long after calcification is well established. So we let the mice age to 6 months of age without doing anything. And then we start giving them high phosphate for another 5 months. So from 6 months to 11 months of age. And again, we give them high phosphate through daily injection. And again, starting from about 6 months of age all the way to almost a year of age. And same protocol, we give them a pretty high dose of high phosphate. And we have a group of animals where we only give them saline, so this salty solution that doesn't contain any high phosphate. And here are the results. First of all, we did a number of control experiments. We verified that indeed the liver was very important in production of high phosphate. So this is a similar high phosphate liver perfusion experiment that we've done. Here is the amount of high phosphate the liver put out in a healthy, well-type mouse. This is the amount of high phosphate in gray here that is put out by the liver from an ABCC6 knockout mouse. So there is a significant drop in the production of high phosphate from the liver when ABCC6 is missing. And the experiments that we've done here is to re-express the human ABCC6 in the mouse liver transiently. And in doing so, we see that there is actually a restoration, not quite complete because we don't have full expression, but we have a pretty good restoration of the amount of high phosphate that is actually being put out by the liver once the human ABCC6 is expressed in the mouse knockout. So here, this experiment confirmed that ABCC6 plays a significant role in synthesizing and generating high phosphate from the liver. The other experiment we've done is to figure out what happens to high phosphates once we give it to the mouse. And this is simply a curve when we injected the ABCC6 knockout mice with high phosphate, and we look into the blood how much it goes away over time. So this is what we see here. After the injections, the concentration is very high, and very quickly, the time is in minutes at the bottom here. You can see that the high phosphate decays away from the blood. And it returns to about the wild-type levels in roughly 2 hours of time, and return to baseline, what we see in the ABCC6 knockout mice, in about 4 hours' time. So pyrophosphate, once it's injected in the mice, doesn't linger very well, or very long, actually, in the bloodstream of the mice. In fact, its half-life, that means the amount of time it takes for half of the concentration to disappear, is about 42 minutes, which is pretty standard, actually, for pyrophosphate. It's quite normal. The other important information we got from this experiment here is the bioavailability. That means how much of the pyrophosphate that we're injecting in the mouse find its way into the bloodstream. And the bioavailability is only 0.5%. That means the vast majority of the pyrophosphate we're giving to the mice never makes it to the bloodstream. Only a teeny little fraction of it gets into the bloodstream. So that was not necessarily very good news. The other control experiment we wanted to do and verify was to determine if the pyrophosphate levels in the mice is changing over time. And what we're saying over time is as the mice are aging. So we looked at the amount of pyrophosphate in the mice over a period of 12 months, Here 3 months, 6 months, and 12 months. These are in the wild-type mice. And we did the same thing in the ABCC6 knockout mice. You can see there is less pyphosphate in those mice, and it doesn't fluctuate over time. So the good thing is that pyphosphate stays, the amount of pyphosphate in the blood stays pretty stable over a fairly long period of time. So we don't have to adjust. The reason we don't do experiments is if we are going to treat patients eventually with pyphosphate for over a lifetime, do we need to adjust the dosage and the treatment if pyphosphate fluctuates as we're aging? And it doesn't seem to be the case in mice, so we can probably continue treating the mice and potentially patients in the future with the same amount of dosage without having to readjust it over time. So this is where actually, these are control experiments just to figure out what's happening with pyphosphate, or the natural history with pyphosphate in those mice. So these are the results now. And you can see this is the dystrophic cardiac calcification phenotype. And on the right-hand side here, this is again a control where we did a surgery to the mice but never induced the injury. And so this is a baseline. That means that's the essentially no calcification in those mice. This is our sham control. Then we did the experiments. We induced an injury and inject the mice with about 100 milligram per kilo of pyphosphate, and we see that there is no significant difference between these two bars. It's a little higher, but there's no statistical difference. So great. Can we see if we can get the same results with lower concentration? So what we did was we halved the concentration. Instead of going with 100 milligram per kilo, we went with 50 milligram per kilo. And we observed the same amount of calcification. So 50 milligram per kilo of pyphosphate injected into the mice was very effective. In fact, it suppressed entirely calcification in the heart. We halved the concentration again and found the same effect. We halved the concentration again when we found a similar effect at 12 milligram per kilo. We halved it again, and we found again no significant calcification. We were wondering how low we could get there. And in fact, we had to go down to 1 milligram per kilo, 1% of what we started with here. And now we're starting seeing calcification going up. So we know from this experiment, and we inject nothing here, you can see that's a normal calcification in this white bar. So what we can say from this experiment here is that the effective dose to inhibit fully calcification in the heart, at least, is somewhere between 1 and 6 milligram per kilo, which is very, very, very low. Considering particularly that the bioavailability is only 0.5% of what we inject. Now, the calcifications that is in the skin of the mice, in the vibrissae of the mice, which is the human analog of PXE, and this is the results. So when we inject the mice for 5 months, right after birth, every day with pyphosphate, this is what we obtain. So the white bar here is our control mouse. If you remember, we inject them with a saline solution, which has no pyphosphate. So this is the normal amount of calcification you would see in an ABCC6 knockout mice. And then when we give the other mice pyphosphate daily with injections, this is the amount of calcification we obtain in the black bar here, indicated by this red arrow. And by comparison here, we have a healthy wild-type mouse. So this is actually a mouse that has no calcification. So when we injected the mice with pyphosphate, we obtained the same amount of calcification as the wild-type mouse, which indicated that there was actually complete suppression of calcification with pyphosphate. The other group, if you recall, when we injected the mice starting at 6 months, where calcification was already well established, and we inject them again daily for a further 5 months, from 6 to 11 months of age. This is the saline group. You can see calcification has progressed from 5 months to 11 months of age. And in the mice we injected, calcification pretty much stayed at the same level as 5 months of age here. So again, we're seeing that, and this is a comparison for control. And then we can see here again that calcification was entirely suppressed in the pre-onset group from 1 to 5 months of age, and was also stopped completely from developing further in the mice that were already 6 months. So that indicates that we can stop calcification from occurring altogether, or we can stop calcification from developing further. The other information we got from this experiment is that calcification had not regressed. There is no reversal of calcification. That was an important thing. We can stop it entirely, but there's no reversal. Of course, we had to look a little more specifically. We had to get the tissue out of the mouse and look for the calcification. So these are these experiments. So this is the pre-onset group. You can see these are the whiskers to the capsules, and this is the calcification. This brown reddish color here, and this is zoom out on one particular vibrissae. You can see this calcification around it. This is the control mice that receive no pyphosphate, and this is the mouse that received pyphosphate, and indeed we found zero calcification. In fact, we had to dig very, very deep in the mice to find teeny little traces of calcification. So it's essentially gone, although there is a little bit still going on, but it's essentially gone in these mice. So this is the second group, the post-onset, when we started injecting pyphosphate at 6 months of age, when calcification was already well established in these mice. The mice that received no pyphosphate had lots and lots of calcification, as you would expect. And the mice that received pyphosphate here had still some calcification that was already there before we started, and it never really developed any further than this. So what we saw from the previous graph was essentially mirrored in these histology experiments, and we quantified those images here, and we found similar results. In the pre-onset here group, from 1 to 5 months, there was a complete suppression of calcification, and the other one, we stopped calcification from occurring further, but it doesn't reverse the established calcification. So the experiment we've done, we've decided to inject the mice, as I mentioned, with a syringe daily in the mice, but my collaborator, András Váradi from Hungary, he decided to go with another route. He wondered whether we could approach this problem in a much simpler way than injection. Injection is great to prove the point, but it's not really something we could do on a daily basis with human PXE patients. That really not be very practical. So we wondered very quickly, at the same time as I developed my experiments, whether we could actually give the mice orally through drinking water. And back then, when we discussed these initial sets of experiments, what we knew was pyphosphate was quickly degraded by phosphatase in the gut. So it was considered there was no oral absorption. So if you drink pyphosphate, it would be lost entirely into the gut, to the microbes in the guts, and would be no absorptions into the bloodstream. But András Váradi being András Váradi, that means he's a good scientist, and what we taught as scientists is to question everything. And he did it anyway. Back then I told him, no, this is not going to work, and others told him, no, this is not going to work either. But he did it anyway, and was persistent. And sure enough, and he presented this in his webinar a few weeks ago, it worked. PPI that is administered orally in the mice reached the bloodstream and is effective against calcification. It is not as effective as the injection, but it's still very, very effective, and it's an excellent use. So, compensating the PPI deficit inhibits calcification development. So here, what I've showed you, this is a summary of the results that we've published. I showed that the reduction in the PPI deficit in the mice is very effective at suppressing the development of calcification, though there is no regression of existing calcification. So we can stop calcification from developing, but we can't really remove existing calcification with this particular treatment. So it works very effectively, but it's not perfect. Pyphosphate is bioavailable, as I mentioned earlier. So the amount that actually ends in the bloodstream from injection is about 0.5% in mice. And from drinking water, this is the experiments done by Dr. Váradi, it's a little less, it's 0.1 to 0.2%, both in mice and human, and that is still very sufficient to suppress calcification. Just to give you an idea of the bioavailability of things that we take commonly every day, such as aspirin, we take a pill of aspirin and its bioavailability is 80 to 100%. Most of it is actually absorbed into the blood. That compares a little bit with the bioavailability of pyphosphate, which is not very good in comparison. So the half-life of aspirin in the blood is 15 to 20 minutes. The ibuprofen, which is another common drug we're taking, has also an excellent bioavailability when taken orally with 100%, and its half-life in the blood is a little longer, 3.5 hours. Zolpidem, which is has a commercial name as Ambien, it's a sleeping pill. It also had a pretty good bioavailability of 70% and just under 3 hours of half-life in the blood. So you can see comparison with these numbers here and what we get from pyphosphate. It's not so good. However, pyphosphate works very efficiently. It doesn't take a whole lot of pyphosphate to be efficient. So small amounts or even intermittent delivery are very, very efficient at suppressing calcification, and that's the good news. Though, these questions remain here. What is a clinically acceptable delivery method? I mentioned that we had these two dual experiments with Dr. Vardy and myself, when I decided to go with injection and he decided to go with drinking water. And injection is not something completely acceptable for lifelong treatments, and daily injection is not something you want to do necessarily. So a drinking aspect or drinking pyphosphate may be a better alternative, but we still have to figure out what is the best methodology from a clinical standpoint. But perhaps there can be another suitable approach, which is a dietary intervention. And I'll explain here. This is something that I discovered about a year and a half, 2 years ago, about a year and a half ago, accidentally. And this is a typical science discovery. It just happened completely unexpectedly. So this is a timeline of events that led us to this discovery. So in the middle of 2017, I put a student, a master student, on a project to study cardiac calcification. And I described to you now. And that student came to the lab, had to be taught all the methods and the techniques. And several months later, in September of 2017, that student was still unable to reproduce calcification in the mice. Her experiments never led to any significant calcification. So at this point, I was wondering whether I was going to change students or I was going to change method. But something needed to be done in order to get this work going. More or less at the same time, Dr. Viola Pomozzi, who was a postdoc in my lab back then, and she was very experienced. She had done this kind of experiments before. Herself also had difficulties reproducing cardiac calcification. So it took us several months to put 2 and 2 together. And we stopped everything and we decided to brainstorm, figure out what would be the problem. And so we developed a possibility, a hypothesis, that perhaps there were pyphosphates somewhere in our system, in the mice, whether in the food or in the water, that would explain why we have no calcification. And sure enough, the following January of that period of time, in January 2018, we measure significant amount of pyphosphate in the food, but not in the water. So the data was come from in Hungary, and we were pretty happy that we found elevated pyphosphate in the food, even though this was a problem. But next is, we were wondering where is this excess pyphosphate coming from? So here we had to do a little bit of an internal investigation. We had to go back in time all the way to February 2015 to realize what really was happening. Because this experiment, this cardiac calcification, was working really well before, and now it was just not working anymore. So we realized that LAS, which is our laboratory animal services, had changed supplier from our institutional animal food, the chow. Back then, they said, we're going to change supplier, we're going to buy from these people because it's a little bit cheaper than from this company because it's more expensive, so we're going to change to a different company. And they, of course, consulted everybody in our institution, and they say, this is the old diet that we're using from this company, and we're going to switch to this diet from this company. And of course, you look at the composition of the various elements, whether it's cholesterol or proteins levels or fiber levels or vitamin levels, and it was all the same between this old diet and the new diet. Of course, from my standpoint, I looked at that part here, which is the how much calcium was present, how much phosphorus was present, how much magnesium was present in the new diet. It was the same amount because these elements are important for calcification. And there was no difference between the old and new diet. So I didn't pay any more attention and I said, fine, no problem, we can switch diet. It turns out that the new diet these people purchased for our institution had a 7-fold increase in pyphosphate over the old diet. And that was a tremendous difference. But we were still wondering whether this could be the source of our calcification problem. So we did a bit of a back of the envelope calculation back then, assuming how much food mouse consume every day, we assumed a bioavailability of about 0.5% from the work we've done, and we calculated or estimated that a mouse would consume about 6 nanomole, which is a unit we used to measure the amount of pyrophosphate here, 6 nanomoles per day of pyrophosphate with the new standard diet. And that's here in this graph here. You can see 2 different sets of measurements obtain about 6 nanomole per day. That would be the amount of pyrophosphates this new diet is providing to the mice every day. We compare this to the minimal effective dose to suppress cardiac calcification that we determined before. One of the graphs I showed you actually showed that information. And we determined that value to be 1.4 nanomole a day. And you can see that the amount of pyrophosphate the mice were getting every day largely exceeded this minimal effective dose to stop calcification in the heart. To confirm this data, we actually obtained samples of the older diet, and then we measured the amount of pyrophosphate. And the daily amount of pyrophosphate the mice would get from this older diet was actually well below this red line, which is a threshold. So yeah, it looks like indeed perhaps the pyrophosphate from this food was actually causing our problem. And you can see there is hardly any pyrophosphate in the water here. So it was probably coming from the food. So in order to prove all of this, we actually went back to the data we had already generated. So we went back to our archive and started comparing the amount of calcification we could see in our mice, in our experiments, before the dietary change and after the dietary change. And we started putting these together into various graphs, and one of them is here. And you can see this is in white, just a control. This is a wild-type mouse. You can see that before there was a dietary change, we had this amount of calcification in the heart. And then once the new diet arrived, we were only obtaining a much, much lower, about a quarter or less of the amount of calcification we had before. So this looks like indeed was the source of our problem. We also looked at this vibrissae, the skin calcification. You can see here the calcification in these images, but this is the quantification. So we looked in mice that were 6 months of age, and we looked at mice also that were 12 months of age. And in both cases, you can see the gray bars is showing that there was about half the amount of calcification now that developed in the mice that were fed the new diet as compared to the old diet in black here. So again, the new diet was also suppressing calcification in the skin of those mice. The next question we asked was, is this dietary pyrophosphate readily absorbed in the mice and in humans? So is it bioavailable? That's essentially the question we asked. And again, we turned to our archives to look at data and started comparing what happened with the amount of pyrophosphate in the bloodstream of the mice before and after the dietary change. You can see here in the old diet in black here, we had this amount of pyrophosphate in the bloodstream. And then when the mice started eating this new diet, you can see there was a significant increase in the amount of pyrophosphate. It's actually a doubling in the amount of pyrophosphate into the bloodstream. So it looked like indeed the mice were able to absorb pyrophosphate through their food into the bloodstream. But you know, as scientists, we need to control and control and control again. We need to really cross all our T's and dot all our I's. So we did further experiments. We had stored samples, blood samples, plasma samples from before and after the dietary change, and we sent this to Thomas Jefferson University to have someone else controlling our own experiments. And this is what the data actually came back to us. You can see in black here the amount of pyrophosphate that was in the mice, in the bloodstream of the mice before the dietary change. And with the red arrow here, you can see there was essentially a doubling of the amount of pyrophosphate that was actually finding its way into the bloodstream of the mice. And we even observed this in wild-type mouse. There is not quite a doubling in these mice, but there was a significant increase as well. So it looked like indeed this was the source of our problem. And from that point on, we realized that if pyrophosphate is indeed absorbed from the food into the bloodstream of the mice and it suppresses calcification, can this also be true in human? So we haven't tested this in PXE patients, but we tested this, of course, with volunteers, and this was done by András Váradi in Hungary. And this is the protocol we use. We found essentially healthy, volunteered students that were roped up around the lab, and András Váradi fed them boiled potatoes and essentially sprinkled pyrophosphate on top of them like you would sprinkle salt on boiled potatoes. And he fed them boiled potatoes and essentially started measuring the amount of pyphosphate in the bloodstreams in the minutes that follow. And actually did this experiment over 4 hours after eating the boiled potatoes. And you can see in this curve here, there is a significant rise in pyphosphate that decreases over time, very much like we've seen in mice with water. And the bottom graph here, you can see these inter-individual differences. There were 8 volunteer individuals that ate potatoes. There's quite a bit of variability between individuals, but it's the same curve pretty much for everybody. You have a rise in pyphosphate within an hour or so, reach a peak, and then start decreasing over time. So this experiment is showing us that indeed pyphosphate absorbed with solid food is actually available in the bloodstream of the patient, in the individual, sorry. And the bioavailability we're getting is about half, actually 0.05%. So it's even less than what we've seen in water, about 4 times less than is available from water, but it's still effective, as we can see from the results in the mice. So in summary, PXE is still an incurable disease, but we're really getting there now. There are symptomatic treatments that probably many of you are aware of, treating with anti-VEGF injections in the eye, preventing ocular hemorrhages. That treatment exists and is being applied to PXE patients. There's been a recent clinical trial with etidronate, which is a pyphosphate analog. That trial gave encouraging but mixed results. It's great, but it's not so great. There are some aspects of the results that were not particularly encouraging, but the other aspect was encouraging. So it's sort of a in-between type of results. There are several other potential approaches we've worked on over the years in humanized mouse models for phenylbutyrate, which is an existing drug that was repurposed to treat PXE. We've been working on this, and I haven't had enough time to speak about it today. PPI supplementation, so pyphosphate supplementation, these are the results I just mentioned, and perhaps in the future we can have genome editing technology that could help further. But for the time being, dietary intervention could be a way to mitigate or slow the progression of calcification in PXE. That's something that could be done. It doesn't need clinical trial or anything like this. So that's a possibility. Pyphosphate is regarded as safe. The World Health Organization says pretty much that you can absorb 70 milligrams per kilo per day without any problems. And in our experiments, we used similar concentration. We went from 50 to 100 milligrams per kilo per day. The Federal Food and Drug Administration and USDA also labels pyphosphate as generally recognized as safe. In Europe, it's actually a food additive labeled as E450. So generally, pure PI, because it's so safe, it's widely used in the food industry. In fact, as a preservative in canned seafood, in baking soda, in cured meat, etc., even in toothpaste. Some toothpaste actually has pyphosphate to prevent plaque development in the teeth. So if this is an incentive to brush your teeth, probably you should brush your teeth even more than you do now. So pyphosphate, these experiments is showing that our experiments are showing that it's readily absorbed in mice and human from injections or from oral delivery, whether from water or from food. And it reduced effectively calcification in mice. So this suggests that perhaps dietary preference also could contribute to the viable manifestation of PXE. As you probably know, there is quite a bit of viability in the presentation of PXE in patients within family or other. Some patients are more severely affected than others. And the areas of calcification, whether in the eyes, in the skin, or the blood vessels, also quite viable in PXE. So the results that I just presented perhaps would suggest that dietary preference could contribute to this viable manifestation of PXE. But there is a but. We're not done. It's very encouraging what we've done so far, but there's still problems to solve. PPI is an effective way of stopping calcification, but if we start using it as a drug, regulations in many countries in the world, including the US, this will become no longer a food additive, but it will become an official medicine. That means you start being regulated by the FDA and will impose a lot of restrictions on how this is going to be used or produced or anything like this. So this is a a significant problem. If we are going to use pyphosphate as a medicine, it will be regulated and we'll have to go through a lot of hoops and regulations in order to have a treatment, an official treatment for PXE patients. How do we deal with this? This has yet to be addressed. The other important things is what are the doses and the frequency of delivery. I've shown high dosage, 50 to 100 milligram per kilo, daily injections or absorbance daily can reduce the potential side effects. All the human volunteers that have been drinking water or eating boiled potatoes are reporting some minor side effects, but there are still potential side effects. Pyphosphate is a salt, that means it contains sodium. How much sodium are we willing to give to a PXE patient? Sodium can increase blood pressure. So what is really acceptable? We have to define that before we use pyphosphate as a potential treatment. Pyphosphate can potentially affect calcification in bones, particularly in children. So can we use pyphosphate safely without affecting the bones? That is a question we need to address. Importantly, how are we going to reverse existing calcification? Apparently, pyphosphate does not address this. At least our results is suggesting that we can't reverse calcification with pyphosphate treatment. So we need more animal work. We will need clinical trial, phase 1, phase 2, when we test the safety of pyphosphate. We need to do this in randomized fashion, and we need to do this in with placebo-controlled trial as well. So this is important. And more importantly, of course, is we need money to do this. And this will require a lot of money to do further animal work, but particularly to do clinical trial. And on the good notes here, there is now an international PXE consortium that involves 4 European countries. Finland, as you can see, number 1, France, Netherlands, and Hungary are all involved in this, and I'm also participating in that consortiums from Hawaii here in the number 6. So there is an international consortium that has been created to tackle those various questions that I just mentioned. And I'll stop here with the acknowledgements, and this was also, of course, done by a team of people. It's just not me working in the lab, but lots of students have come through, and there are some of them are listed here. Chris, Jana, and Viola are being instrumentals, but all the other students are listed here, who are contributing significantly. I mentioned collaboration with András Váradi in Hungary. Is actually on the upper right corner here, and his team have been working also with collaborators in France and the Thomas Jefferson University, Kuhn van the Mediterranean floor. And I'll stop here. Okay, I hope this was all clear then.
Thank you.
All right.